Good evening, everyone. Welcome back to the class on epigenetics. And today we will learn about X chromosome inactivation. And we will just have a quick review. We have discussed about the global or global genome methylation, demethylation activities during formation of gametes as well as in the early embryos. So in the primordial germ cells, we may have something like this, the methylation status. All genes are kind of methylated except for those imprinted non-methylated genes. Somewhere between primordial germ cells to formation of mature gametes, there is erasure of all global methylation and then re-establishment of new wave of methylation. And during fertilization, that undergoes demethylation, one rapid for the paternal or male pronucleus and gradual for the female ones. And some genes will remain methylated, and that is for those imprinted methylated genes. And supposedly, Stella is something, a protein that apparently protects the imprinted genes from demethylation. Okay. So we also discussed about how the problems with the nuclear transfer ones, that is the methylation status for a normal embryo in male and female pronuclei, you will find that there is a lot of methylation, demethylation. Whereas in nuclear transfer, that's not completely demethylated, and then from here and of course de novo methylation happens, and probably they could pose some problems and maybe may result in developmental problems as well. So the activation of zygote, I think we already discussed this too, and if not, there are zygotic genome activation, which involves activation of the egg that has been dormant for so many years. It is filled with nutrition and everything, and all it requires is to um, kickstart the metabolism so that it can undergo cell divisions and then eventually differentiation and so on. There are about 5,000 genes that are expressed right after this zygotic genome activation, which will also include that of metabolism as well as cell division and so on. Today, we will discuss about X chromosome inactivation. First thing is we know that X chromosome, as per the dosage compensation things we have discussed before, in females, there are two X chromosomes, and one of it should be inactivated and eventually forms a bar body. In males, however, that's not the scenario. I hope you remember. I think I gave you a question on that in CIA. So for X chromosome inactivation to happen, there is a long non-coding RNA, which is encoded by a gene called ZIST or XIST. And that is very long. Actually, it's several kilobases. And when it is produced, it will bind or coat to the a center, which is known as X chromosome inactivation center, and then that region will become silenced, and eventually that silencing will spread throughout the chromosome, and there will be a lot of other modifications including histone methylation, H3K9 and so on, and also hypoacetylation. So the lack of acetylation will cause the expression from that region to be suppressed, and further compaction will happen because of histone methylation and so on. And there are also other changes such as recruitment of macro H2A. It is a variant of H2A histone. Okay. So when we have a chromosome and it can undergo the X chromosome, although it forms bar body, it still undergoes replication, because a cell that is present should replicate, and DNA should replicate and cells should divide. And if that is happening, then the X chromosome, the inactivated one, also should get replicated and segregated to the daughter cells equally. And that will happen based on the maintenance of this histone modifications, and also the cyst RNA production as well. Okay, so if we delete cyst or exist, then this X chromosome inactivation is hampered. So one of the limiting factor is exist. As we go further, it's important for you to notice or realize that it is a complex process, X chromosome inactivation. Not every model, which is most likely, but we need to have more evidence. This picture I have taken a long time ago. I took it from a paper a long time ago. Maybe the current research is still there, but we have to wait for the information to come to the textbooks when uh, all the models or every step of it is understood beyond doubt, and then you bring it to the textbook and we study it as such. And when we go to the level of research, there it is uncertain. We don't know what is right and wrong. We are trying to find out and prove something right or prove something wrong. So cyst is a long non-coding RNA. It can bind. We have seen this figure before, which binds to a protein complex is called polycomb repressive complex, complex 1 and complex 2, and they can come here, and then the histones that are present here get modified, because of which silencing happens. There is another aspect to this. X chromosome inactivation. X chromosome inactivation is important, will act on X chromosome inactivation center, and then it will result in inactivation, right? But the expression of cyst itself is dependent on another gene called T6. Please understand that inactivation requires multiple proteins or the RNA probably, and which may be coded by the X chromosome or some other chromosomes as well. So cyst is the component that is mediating or executing the inactivation, whereas T6 seems to be the regulator, or it regulates the cyst. Okay, it is complex, of course. There are complex events. I'm just trying to put it in a very simple way. So this is cyst gene, and here we have T6. So they are encoded by genes that are overlapping on opposite or complementary strands. Here are some of the examples about how T6 could be regulating cyst. One of them or more of them could be right. So if we think of cyst and T6, this is cyst, and you're saying T6, then cyst RNA will be produced, and T6 RNA is also produced. They both can be complementary, isn't it? Because for one of this strand, the lower strand could be acting as a template. For another one, the top strand could be acting as a template. So you are actually getting complementary strands. So in this model here that is explained, the two RNAs encoded by cyst and T6 could base pair, and therefore they cannot silence. Whereas in this case scenario where T6 is not expressed, because of which cyst is produced. Cyst binds to protein factors like that, and they would bind to the X chromosome inactivation center and mediate the repression or silencing of the X chromosome inactivation center. And then that silencing is spread throughout the chromosome. There is another possibility and a model. Some people think this is the way it works, where T6, when it is expressed, it's because you will have two RNA polymerases. One RNA polymerase going in this way, transcribing one of the strands. Another RNA polymerase is going in this way. If this one, the transcription of T6 is high, say at one point there will be a repression, there is an interference between, there is transcriptional interference between these two, because transcription is happening in two opposite strands in the same locus. Then one of it has to fail. So that is one of the ideas. Some people think that is how ZIST is probably um, prevented from expression. But in cases where ZIST is silenced, then you will have production of T is exist, and then it might be um, mediating the inactivation. There are also other ways. People think that there is a protein factor that could be produced because of T6 expression, and that could repress exist. So these are some of the ways people think, and it could be more complex than this. But what you should know or should remember at this point is T6 has an influence on ZIST. Okay, and ZIST is responsible for to acts on or binds to XIX and mediates chromosome inactivation, X chromosome inactivation. So if the expression of T6 is high, you will have inactivation is less or it's not there, because ZIST will be less in concentration or active. So here I would want to show the regulatory network. Don't be alarmed or worried about the so many factors. The expression of ZIST is under positive regulation as well as under negative regulation. There are so many factors here, REPA and ZPX and so on. Don't be bothered about it. What I want you to, at this point, remember is this expression of ZIST can be induced to express, like in positive control, where REPA is involved in increasing the acting as an activator for expression of ZIST. There is also another way where the expression of ZIST could be repressed, and that is by XITE, which will activate the expression of T6, and T6 could act by interfering with REPA, or it directly by interfering with the transcription of ZIST itself. So anywhere we have, this is still a minimized or a kind of bare skeleton gene expression or the network of regulation. There could be many more genes and proteins, genes or their products involved in this. But we will be concerned with ZIST and T6. ZIST is what will bring about X chromosome inactivation, and T6 is a counter or opposite to that of action of ZIST. Okay. There are also some other some things we need to remember is that, sorry, I will first take this and then I will go to the slides. Okay, I will talk about this one. We know about blastocyst. From zygote, zygote divides, forms morula, and eventually to blastocyst. Blastocyst is the first stage where you will have first differentiation. It has the trophoblast cells, that is the outer ring, and then there is the inner cell mass, right? ICM. If you look at the X chromosome inactivation, and all this happens only in females. Males have only one X chromosome, so there is no issue about it. But if you look at the trophoblast cells and ICM, there is a difference in terms of X chromosome inactivation. In the trophoblast, so, well, let me get to this again. The female has two X chromosomes. One is inherited from the mother, and the other one is inherited from the father, paternal and maternal, right? In the trophoblast cells, it is always found that the XP, that is paternal X chromosome, is inactivated. And in the ICM, it's usually, or after ICM giving rise to an embryo, you will find that it is random X chromosome inactivation, which means that some parts of the body could be having paternal X chromosome inactivated. In some parts, you may have maternal X chromosome inactivated. So it's a kind of mosaic images before. So the extra embryonic tissue, especially the placenta that is coming or contributed by the trophoblast cells, you will find that the paternal X chromosome is imprinted or inactivated, whereas in the embryo or the body of the fetus, you will find that it is a mosaic where some parts have a paternal X chromosome inactivation, and in some other parts you have maternal X chromosome inactivation. So the question is, what makes it that the paternal X chromosome inactivation happens first, and why does it happen? So some people will think that it is a pure de novo inactivation. That means when they form the X chromosome, they probably and during the zygote, the paternal X chromosome is reactivated. So in the it has some methylation signatures, which makes it more kind of prone to inactivation. And during the formation of zygote, some people think that the X chromosome is reactivated, and during the differentiation that happens, the first differentiation during blastocyst, they think that in the blastocyst, newly paternal X chromosome is inactivated. Okay. And in the ICM, here as in the zygote, both are active, right? Maternal and paternal. One second. Yeah. So that is what is meant by de novo inactivation. After changing, the inactivation occurs. There are also other people who would say that there are marks on the paternal X chromosome with pre-inactivation markers, because of which it will retain the paternal X chromosome inactivation imprint here in the trophoblast cells, whereas in the ICM, the X chromosome is reactivated and then followed by random inactivation. Okay. It will become clearer, or what I would want you to remember in the next slides is this, and we will also see in the next slides as well. In the trophoblast, inactivation of XP, right? I was discussing in this slide, and during the blastocyst stage, the trophoblast cells, you will have paternal X chromosome inactivated. That is because of increased expression from the maternal allele of T6. If you remember, if T6 is expressed in higher concentration, that will stop the expression of exist, and thereby it is stopping the X chromosome inactivation. So in the X maternal is active, it will remain active, whereas in the paternal X chromosome, ZIST is expressed, which means T6 is not expressed, because of which ZIST is expressed. Therefore, XP is inactivated. Paternal X chromosome is inactivated. That continues as more and more ZIST is produced. It will coat the X chromosome inactivation center, and followed by H3K9 methylation, and then formation of heterochromatin or silencing, and that keeps on maintained in the following cells. In the primitive ectoderm, there is random X chromosome inactivation that we are trying to see here. It is in a given cell, when you have two X chromosomes, in some cases the maternal one produces the ZIST. In some cases, the paternal X chromosome produces ZIST. Whichever produces, that X chromosome gets coated with the cyst and gets inactivated. Therefore, it is actually random. Okay, and that is maintained from the cells that are derived from that particular cell. This should be like a conclusion, like a trying to understand the X chromosome pattern during the whole embryogenesis. So we will start with somewhere here, primordial germ cells. During that time, all of the methylation is erased. We are trying to integrate what we have learned about the methylation status within the global methylation status as well. During primordial germ cells stage and somewhere between formation of gametes, everything is erased. And we don't know, some of the imprints may have already been there. And that forms a zygote. And in the zygote, you have two X chromosomes active, maternal and paternal. And by the time it forms a blastocyst, we have two different cell types, trophoectoderm and inner cell mass. In the trophoectoderm, it's always the paternal X chromosome is inactivated. And in the inner cell mass, the paternal X chromosome, if it is inactivated, will be reactivated, or you can say it as it is erased. It is X chromosome reactivation. So the trophoblast that gives rise to the placenta will maintain that the paternal X chromosome is inactivated, whereas in the rest of the embryo, which is derived from the inner cell mass, will have reactivation. So now at this stage, you have assume maternal and paternal both are kind of active, and one of it randomly one of it could get inactivation. In some cells, they will have a paternal inactivation. Paternal X chromosome is inactivated. In some cells, maternal X chromosome is inactivated. I'll get back to this slide. Just hang on, and I want to complete this, and then I will discuss that. So this is what we are trying to understand, how a body that is a mosaic body forms. Mosaic here we are discussing in the light of only X chromosome inactivation. So zygote, both paternal and maternal X chromosomes are active, early divisions, and during ICM, we are not talking about trophoblast here. We are only talking about the ICM and the body that develops from it. In the ICM, we have random X chromosome inactivation. In a cell, I will call this cell A, B, C, and D. In cell A, we have paternal X chromosome is active and maternal one is inactivated. And all the cells that it would give rise to will maintain that maternal X chromosome inactivation and may give rise to certain body parts eventually, like this. If it is cell B, in which the paternal X chromosome is inactivated, in that case, all the cells that arrive will maintain that paternal X chromosome inactivation and may give rise to different other body parts. And that is how a mosaic in terms of X chromosome inactivation, whether it is maternal X chromosome inactivation or if it is paternal X chromosome inactivation, will depend. Okay. So I want to come to this slide because I have a few questions and I would want you to look at it. And we also had a question based on this as well, theoretically. I want you to find it out. Are there any imprinted genes on X and Y chromosomes? First thing, if there are imprinted genes, they should be, they could probably be, there must be common genes between X and Y. Y chromosome is a shrinking chromosome. People think, or sometimes at some point, they used to say that Y chromosome is becoming shorter and shorter, and eventually there will not be any males. That's not exactly going to be true, but yes. So the X chromosome, if there is imprinting, it should be, the gene should be common between X and Y, right? Only then probably imprinting could make sense. But yes, there could be other possibilities as well. But it's worth trying to find. There are some examples where there are imprinted genes on X and Y chromosomes. I also wanted to, I read this, there was an article or newspaper article at one point, and also there are a couple of research papers which say that intelligence is inherited from mother. Mother contributes, well, you can try to find out what it is. This has no direct relation to what we are, the chapter, but it is an important question to discuss. Mother contributes two things. One of it is mitochondrial DNA, and the second one is X chromosome. Of course, father also contributes to X chromosome in females, right? So if there is any significance, this should be the difference between mother and father, right? And if they are contributing anything special, it probably should have to be with these two, mitochondrial DNA and the X chromosome DNA. Of course, mother is the first teacher, and we are talking if there is any genetic basis for intelligence, and then is it from the mother or is it from the father? And if it is from the mother, and you say you are generalizing it as if it is how it happens, then the limiting factors could be the mitochondria or the X chromosome. Could be. Okay. Um, and yes, that is one thing I think it's worth thinking about. There is another question I have is there any expression from the inactivated X chromosome? If you are saying an X chromosome is inactivated, either maternal or paternal, is it completely repressed, or is there any gene expression happening from the inactivated X chromosome? There are a few examples or publications. We don't know. We may have to wait for validity of it or independent verification of it to see if there is any gene expression from the inactivated X chromosome and what is the significance of it. And the last is the most important one. What I would want you to is find out the molecular mechanisms of X chromosome inactivation. What we are discussing is a topic that is not necessarily mentioned in our syllabus, but it is the basis of epigenetics, is the basis of stem cell biology. And when we are discussing it, there are many things we know and many things we don't know. There are some things we know and many things we don't know. And I would want you to, I will forward you a post and on the Google Classroom. I would want you to check it out and see if you want, if you can understand that literature. Right now, because of this, the course so far, you are in a good situation or position to understand scientific talks on this, on epigenetics. So I would like you to, I will forward the post and please have a look at it. I already shared with you the Coursera course link videos. You probably can watch it and learn more if you are interested to take it ahead as a part of your research. And what we are discussing or why we are studying all these things is the technology, and we are talking about stem cell biology, and the goal is to somehow provide to some um, cell-based approach to treat a patient. And we cannot use embryonic stem cells as much because they may not be compatible with the individual there. So we would want to take we want to use we want to reprogram some adult stem cell into an embryonic stem cell then be able to derive any cell type we want and then put it into the patient. That's the goal of it. So a patient could be maybe having a genetic defect because of which the patient is suffering. So we could take biopsy, we can take adult cells, and then we can use several methods that we will discuss in tomorrow's class, or we'll have a brief introduction today itself, and try to use reprogramming. By there are multiple ways. This is a genetic way of doing it called as OKSM, which would stand for OCT4, SOX2, CMYC, and KLF4. These transcriptional factors are responsible for pluripotency. They are pluripotency inducing transcriptional factors. So sorry, if we genetically overexpress them, the cells that were adult stem cells like this could become induced pluripotent stem cells. And if there is a genetic defect, we could probably, using recombination or recombinering, we can repair the genetic defect and then derive whichever cell type we want and then transplant into the person. We can also use the same cells for testing in drug discovery about compounds for the toxicity or effectivity. The way how are we different from the doctors? If you ever wanted to become a doctor and you didn't, so this part of biopsy from a human or transplantation of the corrected cells into the human, only a qualified doctor should do, a licensed doctor. So in the Western countries, usually the doctors also perform research. In our country, it's not possible because of the facilities and also the amount of demand we have. We are highly populated. The doctors here, only majority of them treat, and they're not involved in research as much. That's sad, but it's not possible either. So if you are planning to become a researcher or a scientist, this is your domain. This becomes your domain. That means your ability to produce induced pluripotent stem cells, make corrections to the genetic defects, differentiate, and we get the cells from the doctor. Doctor takes biopsy and he or she takes the cells, and we give back the corrected cells, and the doctor will transplant them into the human body. In the lab, as a proof of principle, people work with mice or guinea pigs and so on, where they're in that case, you may sometimes, if it is a larger animal, they rely on veterinary doctors. But if it is mice and other things with uh, simple um, training, some about animal handling, we can also do those things. Okay. So but this is a huge area of research. All the things that doctors will usually eventually use is comes from the researchers, and we could, if you wanted to be in research, and these are some of the areas where you could be working in association with doctors. So we have uh, I'll just take 5 minutes and I will stop. So what we have learned is, or why we learned so many of these epigenetic modifications, is because we want to reprogram. This is the area we want to reprogram, make embryonic stem cells or pluripotent stem cells, and then make them back into differentiate them and be able to replace an organism or so. And for that, we have the issues that we have are the epigenetic modifications. We have discussed about DNA methylation, histone modifications in greater detail, and also X chromosome inactivation, global DNA methylation, which also comes in that way. So if we want to make reprogram to make embryonic stem cells or induced pluripotent stem cells, then we need to consider all these things. The X chromosome reactivation, if it is inactivated, if it is from a female, and then we should be able to reactivate it. We should be able to check on the demethylation methylation status, and we should hope that the imprints are intact, because we don't want those imprints to be, if we are taking a somatic cell and then we are hoping to make it as an embryonic stem cell or induced pluripotent cell, then the imprints here could, majority of them are in methylated state because they are no longer used for the somatic or highly differentiated cell. Okay, it's not that those genes are always expressed. And if we are trying to reprogram, we cannot be assured that those imprints are maintained as such. That is why we are trying to learn about all these things. And we should somehow express the pluripotent genes, which are about OKSM mostly. And the chromosome numbers, every time we do transplantation of nuclei and so on, there is always a risk of chromosome number abnormalities. Aneuploidies may occur. So that is another thing we need to take care of. If we have formed embryonic stem cells or induced pluripotent stem cells, they should be able to form embryoid bodies, and they should demonstrate, or we should be able to demonstrate, that they are pluripotent by their ability to show that they can form all cells from the three different germ layers: ectoderm, mesoderm, and endoderm. So there are, I'll just give an introduction and then I'll talk a little bit about something else. There are, nuclear transfer is something we know. We have already discussed somatic cell nuclear transfer, right? You take the somatic cell from the patient, you put it in, and you will get, um, you can get embryonic stem cells. Those are highly compatible with that of the patient. But then there are problems because the technique is inefficient, and secondly, there are a lot of ethical issues that are associated with this. Cell fusion is you take a cell and grow it along with the embryonic stem cells. This is from patient, this is from somebody else, and you grow along, and you use virus or polyethylene glycol and other things to fuse these cells. This is a cell and this is a cell, and they become one cell. One cell has brought in 2n, and that one other one has 2n. So you actually have a tetraploid cell, which may further divide to give rise to 2n, out of which some cells may demonstrate pluripotent and also be having characteristics that of the patient if they are derived from here. But then the fusion rate is very low, and the problem is the tetraploids. That is a problem. Some people have suggested that instead of taking whole cells, take the cell extract from embryonic stem cells and then grow the cells in this one, in here, and then we will get, we can probably reprogram the adult cell, or a completely somatic cell, into a pluripotent cell. Yes. What I want you to right now look at is, I want to show something else, which is with regard to the integrity. And let's see if it is here. If you are reading about stem cells, and you will read a lot about some of the controversies that are there, in that there is an example, a classic example of this. So people have been trying to reprogram and try to get very efficient or good embryonic stem cells or pluripotent stem cells. And in trying to do so, they have, I hope it will load fast. There is something called as STAP cells. Okay. That means they the theory, at least in theory, it is this: you have you isolate cells, grow them in dish, then induce stress in them. When you induce stress and wait for a few days, apparently the cells become pluripotent stem cells. And there's from those pluripotent stem cells we can derive any type of cells. This is so easy, so fascinating compared to what the other techniques. We virtually cannot see any kind of defect in this, right? Because we haven't added any genetic factors. We didn't fuse cells. We did nothing. All that is, it is very simple because all we are doing is just give stress. But then the problem, this is kind of invented by a person, this Japanese lady, and she published 2 Nature papers or so, some 3-4 years back. As it turns out, nobody in the world could reproduce this experiment, what this lady has published. And eventually, the papers, it was found out that this is all fraud. She has probably fabricated the results. And then, uh, so this is controversial because it is bordering between ethics. First of all, what we are doing is wrong and faulty, and some people might be wasting a lot of resources on your because of your results. And here is one of her ex guide. He guided her PhD or so, and this person in Japan, they take it very seriously, and this person committed suicide in his own lab because he couldn't handle the embarrassment or humiliation of being associated with a faulty or fabricated results. Or probably he believed that this step is right, but anyway, he committed suicide and died. I think it is him, but one of her guides actually committed suicide and died. I hope, I want to leave you with this, with one second. I want to leave you with a message on that lines. To be a scientist, okay, we need many things. We need critical thinking. We need self motivation. We don't need somebody else to come and say, do this or do that, and so on. We by ourselves are self motivated. And the third is lot of energy to perform the experiments. And there is another one called as grit or persistence. Science is, as I described, this is a ocean of unknown. And you're here, and nobody else wants to get in, and you are the one trying to go in there, and you don't know what to expect. And each step you take towards it, you have to find way and try to be able to stay afloat. That means you're doing something unknown for the first, you're finding something from the whole vast ocean of unknown. That means failure is common. How many of us can handle a failure? A simple experiment fails, we feel sad about it. Imagine doing that over days and weeks together and sometimes years together. Failure is the norm of a scientist's life. If you haven't failed, there is a saying, if you haven't failed, you did nothing new. It is something like that. That means one should be willing to fail and still get up and be able to do experiments. For that, you need a lot of you need a big heart or a strong heart. It's not, science is not for everybody. And the last and the most or more equal important to all of them is integrity. Okay, that means we are people of information, and what we say, the information that we get comes out of us to anybody, should be highly valid to the best of our knowledge, right? The information should be valid. As a scientist, you are generating new information from the unknowns. And for the sake of raise in profession, or for the sake of publications, or for sake of salary hike, or climbing the ladder, whatever it be, or name and all, there is always a temptation to fabricate results and then try to move away. And that is wrong for a scientist, or anybody in general, scientists especially. That is why scientists, you can find many scientists all over the world. Not all of them are truly scientists, because some of them compromise with this. For a person to be emotionally strong, to handle the failure, to be able to move forward even though there is a lot of failure, want to have a lot of energy, motivation, critical thinking, and then be sincere about it, that is a difficult life in that sense, and that makes it also worthwhile to pursue as a scientist. And if you, any of you, are practicing to become a scientist, all that is most important is integrity. And that is the reason why I also take integrity very much. You can be weak in everything else, not in this. Okay? Because integrity is something difficult to maintain. It's easy to cheat, it is easy to fabricate. And so in the lesson, or in programming, nuclear reprogramming, there was a huge controversy, the one that I was discussing about. It was a huge, grandly celebrated. You also might have heard about, recently there was an issue with NCBS Institute with integrity of paper. A student, a lab people are involved in fabricating the results and publishing in a very reputed journal. And eventually they were called out, and then the paper was retracted, and there was serious actions taken about. Nowadays, it's very difficult to fabricate. People are there who will keep on fabricating, but they will eventually be caught out. But that's not, we don't need somebody else to catch us. What we need is to be by ourselves integrous and work towards science to generate new knowledge that is useful for the well-being of our planet. Okay? I'll stop here. In the next class, we will discuss a little more about programming, and then we'll go reprogramming. And then these are some of the experiment. I would also want you to, I will talk about this one specially in the next class. Okay? Any questions? Anybody? Okay, if you don't have any questions, we'll meet tomorrow or in the next class. Okay? Take care and have fun.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Bye-bye. Bye.